Hello, everyone. Welcome to the lab practical exam midterm review for Professor Ravikumar's Bio 1 course. This will be for fall 2021. So we got the review document here, and pretty much I'm just going to go through all these questions and tell you which answer is correct, and then maybe a little bit of information as to why. Alrighty, so let's get started. Which of the two variables are utilized in the scientific method? X and Y, positive, negative, dependent, independent, or hypothetical and experimental. For this, it's going to be dependent and independent. These are the two variables utilized in the scientific method. The dependent variable is going to be what you're actually observing, and then the independent variable is going to be what you're changing to see how it influences whatever you're observing. So yeah, if we go down to question 3 here, we're talking about different diets affecting the size of tadpoles. In this case, the diet would be the independent variable because that is what we are changing. And then the size of the tadpoles, what we're studying, so that would be the dependent variable. So actually write down here, what is the independent variable of the experiment? Again, what is the thing that we're changing? That's going to be the diet for the independent variable. And then, so I'll write here independent. And then right here, the tadpole size, this would be the dependent. Let's see. When graphing your data, it is important that you do one of these things. So in this case, this is one of these things you have to sort of just memorize how things are formatted, but you always want to place the independent variable on the x-axis. And then if you have the independent variable on the x-axis, you always want to place the dependent variable on the y-axis. So this is just one of those things, again, you have to memorize. But yeah, it's just very common formatting. So independent variable on the x-axis, the dependent on the y. And then down here, this question, imagine that you wish to compare two different diets that will be fed to tadpoles. One diet is meat-based, the other diet is lettuce-based. You want to see if the meat-based diet will be associated with an increase in the average weight of the tadpoles. And you keep all other factors the same. You only change the diet. So what is the thing you are changing? That is going to be your independent variable. So that is the diet. And then what are you observing? The size of the tadpole. So that will be your dependent variable. So again, with dependent and independent, you're just really trying to see how the dependent variable depends on the independent variable. So, all right, so more stuff about experimental procedure. Number 4 here, if you wanted to test whether or not a particular pain reliever was effective in the general population, you need to design an experiment with an experimental group and a control group. What should be the only difference between these two groups? So it doesn't, first choice, it doesn't matter what is different between the two groups. That is incorrect. If there's lots of differences, you're adding all sorts of different factors that could influence your results. So definitely not the first one. Age, also not a good thing to change between the two groups because, people of different ages can process medication differently, but also that's not what you're studying. You're not studying age. Sex, so here the control group should be all women, the experimental group should be all men. Again, this is just changing another variable that you aren't really concerned with. But then if we go up here to the top of the next page, our last option, treatment. The experimental group should get the pain reliever and the control should get the placebo. This is the correct answer because, what are you studying? You're studying if a pain reliever is effective. So the only thing you want to differ between those two groups is whatever your experimental variable is. So in this case, it's just whether the pain reliever is received or not, because everything else should be the same. You're only studying the effect of the pain reliever. So the control should not receive the pain reliever, while the other group should receive the pain reliever. Let's see, a good scientific experiment is reproducible. What does this mean? I like to think about it like this. So if, and also right here, so, well, actually, I'll explain this first. So if you're given a set of instructions on how to carry out an experiment, no matter who is reading those instructions, they should get similar results if they carry out the experiment the same way. So that is essentially what reproducible means, that if different people, a different place, different lab and everything, if a totally different set of equipment and people are being used to do the experiment, they should get the same result. So, but yeah, the way I like to think about it is like if you're given the instructions on how to build an airplane, it doesn't matter who is building the airplane or where it's being built or anything like that. If they follow the instructions correctly, at the end of the instructions, you should have an airplane. and it should work and do everything an airplane does. And that's essentially the same thing here. If your experiment is written out correctly, where all the steps and details or everything are listed, then no matter who carries it out or where it's carried out, you should get the same results. Yeah, because if people are, doing the same experiment, same procedure and everything, getting totally different results, then that just shows that, yeah, there's something weird going on. So, yeah. But also, so number 6, for an experimental finding to be valid, it must be reported. This is true. So pretty much, if you find out something in your experiment, you need to tell other people about it. If it's true in one case, that's not really, yeah, it could just be a fluke, or it could just be maybe, something happened once. But for something to be valid, you must report it, and other people must also be able to test it out and try it and verify your findings. So yeah, for it to be valid, it must be reported. How is the metric system different from the standard United States system? The United States system is also known as the empirical system, but the metric system is based on units of 10. Let's see, what instrument did you use to weigh objects in the metric system lab? So which one of these deals with weight? Not thermometer, not ruler, definitely a scale, and then not a compass. So dealing with, yeah, weight, scale. In the metric system, the base unit of weight is the gram. So pretty much when you're looking for an answer for this, essentially in Bio 1, you want to look for whatever the unit is without any prefixes. So if you're looking for your base unit of weight, it's going to be gram. Base unit of fluid is going to be liter, because again, there's no prefix. And then base unit of length would be meter, so no prefix. So those are your base units: gram, liter, and meter. All right, when measuring the volume of water in a graduated cylinder, it's important that your eye is parallel to the meniscus. This is true. You have to get an accurate reading on any liquid in a graduated cylinder. You need to get eye level with it, and then take the measurement from the bottom of the meniscus. And you really can't tell where the bottom of the meniscus is unless you're eye level with it. So definitely get parallel to the meniscus. A Biarritz test would indicate a positive result for proteins in which of the following solutions? So Biarritz test, testing for proteins, and then which one of these solutions has protein in it? Not starch. Starch would be polysaccharides. Olive oil would be lipids. Glucose would be monosaccharides. Distilled water has nothing in it, but albumin solution, albumin is a protein. So we would get a positive result for protein in albumin solution because, again, albumin is a protein. So which reagent is used to detect for the presence of protein? We just answered that in the previous question. Biarritz reagent. Which reagent used to detect the presence of starch? That's going to be IKI or iodine or potassium iodide. Any of those names would be accurate, but IKI for potassium iodide. Let's see, which of the following tests it detects for the presence of lipids? That would be the brown paper test. Let's see, and again, the brown paper test, that is where you take a piece of brown paper, you apply different liquids to it, and then after the liquids have had a chance to dry, you check them and see if there is a stain on the brown paper, and if you shine a light through it, if the light can shine through, then that means that there was lipids present. So that is what the brown paper test is. All right, which color change represents a positive reaction for the presence of starch using iodine? So iodine starts as kind of a yellow or amber color, and then it turns into a very, very dark, essentially blue-black color. So that would be a positive result. So a blank solution has the same concentration of water as the cell placed in the solution. So here we're now talking about tonicity. And again, tonicity is all about the concentration of solutes within a solution. So in this case, we're talking about the same concentration of water. So here the keyword is same. So if the water content and the solute content is the same both inside the cell and outside the cell, then that would mean that we have an isotonic solution where, yeah, both concentrations are the same. Let's see, when testing the tonicity of red blood cells, if the solution becomes transparent after adding blood cells, you could assume something. In this case, if the solution becomes transparent, that means that something is happening to those blood cells that is making it so that they are no longer like solid blood cells. So what's happening is the solution is allowing water to enter those cells, causing them to swell and burst and having their contents sort of just spill out so that they're no longer visible. So in that case, it would be hypotonic, because if something is hypotonic, it means that there is less concentration of solutes or salts or whatever on the outside of the cell, which means that the water will be attracted to the inside of the cell, which means that water will again flood into those cells and then cause them to burst or lyse. Lysing or to lyse is the term for a animal cell that bursts. So hypotonic cell bursts. When the Elodea leaves were placed, and again, Elodea is a plant cell. When the Elodea leaves are placed in the 10% sodium chloride, what is the result? So in this case, we're placing it in a very high salt solution, so 10%. So in that case, this would be a hypertonic solution, which means that there are more solutes on the outside of the cell than on the inside, which means that water is going to leave the cell and then be attracted to the solutes in the solution. So in this case, the cell membrane will shrink away from the cell wall because, again, the cell wall is pretty rigid and it won't move very much, but the cell membrane will. So it'll shrink away from the cell wall because the solution is hypertonic, causing, again, the water to leave the cell membrane. Crenate red blood cells can best be understood by viewing them through a microscope. So if blood cells crenate, they will shrink, and they have cytoskeletal elements in them, so they'll pretty much shrink wrap the cytoskeleton. But to actually see this, it's best viewed through a microscope because it's very obvious, and yeah, it's really easy to see if they're crenated just by looking at them. So, all right, 21. If blood cells are placed in a hypertonic solution, what will happen? So this one's kind of tricky. Let's see, if you see the third answer down here, the cell will shrink due to the loss of solutes from the blood cell. That's almost right, because if it's in a hypertonic solution, water is going to leave the cell. So the cell will shrink, but it's not because the cell is losing solutes. This stuff about tonicity is all about the movement of water. So in this case, the cell walls will shrink due to water loss by the cell. So it might seem like it's number 3 here because it says the cell is going to shrink. But again, the key here is that it's not losing solutes, it's instead losing water. So, yep. Let's see, in order to be of scientific value, it's important that a hypothesis be testable. So factual or false or correct, well, factual and correct, I suppose, are similar. But really, yeah, a hypothesis isn't very useful if you can't test it. So that is the main qualification for a hypothesis, that it is testable. Let's see. The hypothesis is the braking distance grows linearly with speed. What is commonly graphed on the x-axis of this graph? So for this, we have to remember what variable goes on the x-axis and what variable goes on the y-axis. So in this case, we are looking for what goes on the x-axis versus the y-axis. So on the x-axis, we have our independent variable. And then our y-axis, we have the dependent variable. So in this case, let's look. Let's see. We're testing, or what we're trying to see if the braking distance is affected by speed. So we're modifying speed to see how it affects braking distance. So here, braking distance would be our independent variable, or sorry, speed would be our independent variable because that's what we're changing. And then braking distance is what we're studying. So that would be our dependent variable. So in this case, speed would go on the x-axis. Because again, speed is what we are modifying, and we're studying to see how braking distance is affected by it. So yeah, speed, independent variable, which then goes on the x-axis. In the pill bug experiment, your hypothesis was related to the environment that pill bugs prefer. So you're trying to see if they prefer, I believe it's sand, or if they prefer what is it? Cornstarch. Let's see. Let's see. The substance used as the control was sand. In the pillbug experiment, an important part of the experimental strategy was to make observations at regular intervals. This last one here, 3 observations at 5-minute intervals, is might seem right, but yeah, it's just make observations at regular intervals. That is the more correct version. Let's see. According to the results of the pillbug experiment, they tend to prefer cornstarch over sand. In the experiment, how many pill bugs did you need to get good information for the experiment? That was 10. Want to make sure your sample size is large enough. And then here we have another question about forming hypothesis. So this one sort of is like kind of like a writing prompt or sort of like a reading prompt. You want to take the information that's given here to figure out what answer is correct. So some students consume large amounts of caffeinated drinks to help them stay alert when studying. You notice that many who engage in this practice seem to do poorly on exams. Suppose you want to investigate the relationship between caffeine consumption and exam performance. Which of the following statements would be an appropriate hypothesis? So, number one, many students consume large amounts of caffeine while studying to improve their exam scores. May be true, but the information we're given doesn't really support this as the hypothesis. Caffeine increases alertness, but also anxiety. This here could be true, but up here, we don't really see anything about alertness or anxiety. So again, this is probably not the best answer to choose. Number three, students who consume large amounts of caffeine while studying have lower exam scores than students that consume less caffeine. This one really is supported by this information that we're given. It says that some students consume large amounts of caffeine to stay alert, but you notice that many who engage in this practice seem to do poorly on exams. So this little sentence right here sort of gives you an indication of which one of these options is sort of your best bet for what a hypothesis should be. So this one should avoid consuming too much caffeine while studying. Another potentially good one, but this one's sort of more accurate here. And again, when you read up here, doing poorly on exams, this one tends to relate more to that. So again, you want to really look at what information you're given. And then too much caffeine is harmful to your health, potentially. But yeah, again, we're not really given a lot about caffeine and health up here. So yeah, this third one here is sort of the most relevant to the information that you're given in the prompt. All right, back to pill bugs. So the pill bug experiment, the bugs move towards the cornstarch. What would you have concluded? That they tend to prefer cornstarch over sand because more of them move towards it. A good scientific experiment is reproducible. This one is looks like a repeat question. So in this case, yeah, you can conduct it anywhere with anyone, and the result should be the same. How many pairs of legs does a pill bug have? That's going to be 7. What best describes the pill bug's organ of respiration? This is a weird one because they're like land-dwelling organisms, but their organs are most resembling of gills. So they have gills. Let's see. How does a pill bug react when confronted with noxious or unpleasant substance? It will move away from it. How is a control group treated in a scientific experiment? So control group and your actual experimental group, you want to keep them exactly the same, except the experimental variable. So just like our previous question talking about pain relievers, if we have 2 groups, the control and the experimental group, what should be the only difference between those groups? Not age, not gender, not any of that stuff. You only want to have the only variable that's different be the treatments. So again, in the example of the pain reliever, we only want the pain reliever to be the difference between the 2 groups. We want the 2 groups to be as identical as possible, except for whatever we're studying. So let's see. Which is not one of the phases in the 4-phase model of the scientific method. That would be discussion. Let's see. In which phase of the scientific experiment or in the scientific method is the strategy carried out and the measurements collected? Let's see. So when it says the strategy carried out, so you're actively doing something and collecting measurements, that sounds awfully like an experiment. So that is going to be the stage when you do that. And then here, it's a repeat question. In order for to be of scientific value, it's important that a hypothesis be testable. All right, now we're talking about the function of buffers. So in this simulation, when you add acid to distilled water, what happens? So we're adding acid, which should lower pH to distilled water. So distilled water is not a buffer, so it is going to easily have its pH changed. So the pH will decrease. So again, because distilled water is not a buffer solution, so it has no ability to resist changes in pH. So if we add acid to distilled water, the pH is going to decrease. Let's see, within the function of buffer simulation, why can you deduce that the artificial cytoplasm is a buffer? So What does a buffer do? A buffer will resist changes in pH. So if it resists changes in pH, and we can deduce that the artificial cytoplasm is a buffer, then we can see that the artificial cytoplasm resisted changes in pH. So yeah, if you add an acid or a base to something and the pH doesn't change, then it has some buffering capacity. So yeah, we can deduce that the artificial cytoplasm, if it resists changes in pH, then it is a buffer. All right, 42. When acid is added to water, the pH drops drastically. This is because water is not a buffer, so it has no buffering capacity. Assume that you have an unknown solution in a test tube. When you add phenol red to the test tube, it immediately turns pale yellow. This is an indication that the solution in the tube is one of these things. So I'll bring up this picture real quick. This is phenol red in its range of colors. And we'll see as we go to the left, the pH decreases. So it gets more yellow as the acidity increases. And then it gets more dark purple as the basicity increases. So if we have a solution, as we have in this question here, we add phenol red to the solution, and it immediately turns yellow, then that would tell us that the solution is an acid. Because, yeah, phenol red is a pH indicator. So if it immediately turns yellow, that means it's in an acid. So it's become very acidic. So, yep, that would let us know that that unknown solution is an acid. All right, 44. In the function of buffer simulation, what do you think would happen to the pH of the artificial cytosol if instead of adding acids, we added a base instead? So there's two parts of this question. It says instead of adding acids, we did the experiment, we added a base. So you may think, oh, the pH would increase because we're adding a base. But the artificial cytosol is a buffer. So whether an acid or a base is added, it should maintain its pH, because that is what a buffer does. It allows us to resist changes in pH. So just look at what you're adding stuff to. So if the question was, if you added a base to water, then the pH would increase. Or if you add an acid to water, the pH would decrease. But if you add an acid or a base to a buffer, then the pH stays about the same. In gel electrophoresis, what is the agarose used for? The agarose is used for making the gel. How does the DNA rate of travel differ for small DNA versus large DNA? Small fragments will travel farther than large fragments because they have to move through that gel, and it's easier for small fragments to move than large fragments. Which of the following of DNA is true? This is pretty straightforward. It is the genetic material of cells. Tonicity in cells refers to the concentration of solute in a solution outside of a cell relative to the solute concentration of the cytoplasm inside the cell. So yeah, tonicity is all about the concentration of dissolved solutes within a solution. So the outside of the cell, the concentration of solutes outside of the cell, versus the solute concentration inside the cell. So yeah, solute concentration outside versus solute concentration inside. A blank solution has a higher concentration of water and lower concentration of solute than the cell placed in the solution. So higher concentration of water, meaning there's more water outside, lower concentration of solute also outside, so there's less stuff dissolved in that water, which means that there is more stuff within the cell, which would mean that the solution outside is hypotonic. Because again, a hypotonic solution has less dissolved solute than the cells that are placed in it. So here it says it has a higher concentration of water and a lower concentration of solutes, which means that there's a higher concentration of solutes within the cell, which again means that the solution is hypotonic. When testing the tonicity of red blood cells, if the solution becomes transparent after adding blood cells, I think we already answered this one, I think. But yeah, if you add red blood cells to a solution and it becomes transparent, those cells have burst. And again, they burst because water has entered those cells, which means there's a higher concentration of solutes within the cell which means that the solution itself is hypotonic, having a lower concentration of solutes. A blank solution has a lower concentration of water and a higher concentration of solutes than the cell placed in the solution. So here, the key is higher concentration of solutes than the cell. So if there's a higher concentration of stuff outside the cell, then that's going to mean that solution is hypertonic. So water will be drawn out of the cell towards that high concentration of solutes. Let's see. Why do you think it's a good idea to soak wilted lettuce in cool water before serving it? So this has to deal, again, with tonicity. So if we have plant cells and we, or if they're wilted, if it's a wilted leaf, then that essentially means that those plant cells don't have very much water in them. Because as we add water to plant cells, they become more solid, because it's essentially like if you have like a water balloon or something. When the water balloon is empty, it's pretty flimsy, but as you add water to it, it has more shape to it, becomes more solid. So it's the same thing with these plant cells. If we can somehow get water into those cells, then it'll become more rigid, a little bit sturdier, or crisper. So we want to place the cell, or we want to place the plant cells in a hypotonic solution. So the water is a hypotonic solution relative to the cytoplasm of the plant cells. So that means that the water will be drawn inside the plant cells, which will mean that they will become more rigid, they'll become more sturdy, they'll become crisper. So if you have wilted lettuce leaves, you can have some water, place them in it, and they will absorb that water and become a little bit crisper. If you were to place this lettuce in a hypertonic solution, then that would mean that the water that is in those cells would be drawn out into the hypertonic solution, so that would make it even more wilted. So that's why it's important that we have a hypotonic. If we did an isotonic solution, then nothing would happen. So equal parts of water would move in and out of the plant cells. So there would still be movement, but yeah, it would be in equal parts. So, all right, 53. When you receive fluid in the hospital, it is often 0.9% saline solution. What would happen if the nurse used distilled water instead? So this 0.9% saline is at this very specific value for a very specific reason. When you're getting fluids intravenously, they're going to be in and around your red blood cells. And for your red blood cells to function optimally, they need to maintain that shape. So we don't want them to crenate or shrink, and we don't want them to swell or potentially burst. So this 0.9% is exactly what we need to maintain an isotonic solution so that they maintain their shape. If we were to instead use distilled water, that's essentially using 0% saline solution. So that would be a more hypotonic solution than this 0.9%, which would mean, since it's a hypotonic solution, that the water from the solution would be attracted into those cells, which would mean that the distilled water would be hypotonic to your blood cells. So the cells would gain water, swell, and possibly burst. And that is not what you want in your own red blood cells. So, but yeah, just essentially, yeah, using distilled water is even less of a concentrated solution than 0.9%. So yeah, it's hypotonic, and those cells would gain that water and then potentially burst. Which metric unit is most appropriate for expressing the size of bacterial cells? Well, for looking at bacterial cells, we typically use a microscope, which deals in micrometers. So let's see. Learning the basic lab safety rules. For those, it's pretty much what kind of clothing to wear in the laboratory. Is it ever okay to eat in the laboratory? Where do you put different types of waste? And all of that sort of general lab safety stuff. There's a few questions about it to give you an idea of the kind of questions that you might see, but really just your general lab safety rules. And then studying parts of the microscope. Definitely just look up labeled microscope or labeled parts of a microscope. You may find this exact same image here. And yeah, you just want to make sure that you know generally what all these indicated parts are. So definitely look that up and check that out. Let's see. So the blank causes the stage to move upward or downward. That is going to be the course adjustment knob. So because if you think about it, you have your slide here on this stage, and the thing that's going to adjust the focus is going to be actually moving the stage up and down. So it's going to be the course adjustment knob. The magnification of the ocular lens, that's going to be these two lenses up top, is generally 10 times. So that's different than the objective lenses. The ocular lenses, again, are here up top where your eyes go, and then the objective lenses are down here near the slide. All right, 56. Pond water can contain many types of organisms. Which of the following are most likely to be found in a pond water sample? So really, we can eliminate a couple of these. So viruses are not actual organisms. So they're not organisms that can be found there. They're also much too small to ever see in a microscope. So, but yeah, they're not organisms. Same for human cells. You're not going to find live human cells in a sample of pond water, but you will find bacteria, which are present essentially everywhere. You will find algae. You'll also find rotifers, and you'll find protozoa. So pretty much everything except for human cells and viruses. What is enhanced when a stain is used to make a sample or parts of a sample darker than the background on the slide? So let's go through these right here. Magnification, that is how large something is. So that is not going to be enhanced by staining something. Contrast is the difference between various structures as far as color goes too. So if you're staining something a darker color than other parts of it, then you are increasing the contrast. Media type, that's not going to change. And then resolution is how much detail you can see. So that's not it either. It's definitely going to be contrast. The total magnification of an image is determined by blank, the magnification power of the ocular lens and the magnification power of the objective lenses. So this really is very straightforward. To calculate the total magnification, you multiply together the magnification of the ocular lens and the magnification of the objective lens. They both multiply together. So yeah, you multiply those two numbers together and you get the total magnification. All right, and then lab safety stuff. In which circumstance is food and or beverage allowed in the laboratory? Never. Which of the following would be the most appropriate for the disposal of cultures on agar plates? So cultures on agar plates, those are essentially bacterial plates that have bacteria growing on them. So that would go in the biohazard bag or bin. That's not for the normal trash, and that's not for the sharps container. Because again, sharps containers are only for materials that are contaminated and are contaminated with biological material, but also could puncture that biohazard bag. So plates are not sharp enough to do that, so they do not go in a sharps container. Which is the most appropriate disposable for a swab used to culture your throat during the lab? So a swab is not sharp, but it does have biological materials on it. So that would go in the biohazard bag. Again, anything that has living tissue or once living tissue or anything that's come from a living organism will go in the biohazard bag. The purpose of lab safety rules and protocol is to protect everyone in the lab from injury and infection, of course. It's not to enable students to complete the lab and activities in less time. It does help keep the lab clean and orderly, but this is the main reason. So, and then when should you wear closed-toed shoes in the lab? Every time, always. Yeah, so just general lab safety stuff. But that concludes all of these questions. Yeah, so hopefully that was pretty straightforward. Another thing to know about your exam, you will have one attempt for each of your questions. So pretty much once you see a question, answer it to the best of your ability, because once you click next and navigate away from that question, you can't go back to it. And that may seem kind of rough, but that is how an actual lab practical goes, where in person you would move from station to station, and typically you'd have 2 questions per station, and you'd have like roughly 2 minutes or so to answer them. So in person, you don't get to go back to questions either. So it's pretty realistic. But just know that once you click away from a question, you can't go back. Also, there's a chance you may receive like pictures as far as like some of those chemical tests go, or you may, regarding microscopes, have a picture of a microscope asking, to label a part or something, or maybe asking to, identify a specific cell type, or maybe like some organelles or something like that. Remember what chloroplasts look like, that sort of thing. But yeah, that's about it. So if you have any questions, feel free to send me an email. But if not, thank you for stopping by and sticking around to the end of the video. So Best of luck on your exam. Hope this was helpful.